This is a production of Cornell University.
Okay, shall we get started?
Sure.
Okay, well, hello everyone. Welcome to our first entirely Zoom seminar of the Plant Biology Seminar Series. It's a pleasure to introduce Adam Boyko. And Adam's an associate professor in our vet school in the Biomedical Sciences Department. He earned his Bachelor of Science degrees in both computer science and ecology, ethology, and evolution at the University of Illinois, Urbana-Champaign. Then he earned, a MS in computer science at Purdue University and a PhD in biology at Purdue. He did a postdoc at Cornell with Carlos Bustamante and moved with Carlos to Stanford. And then he returned to Cornell as an assistant professor in 2011. Adam's lab uses dogs as a model of genetic disease and evolutionary genetics. And I invited him to our seminar in part because many of us are very interested in genomics and the kinds of things he's doing. And also, I invited him as our career-related seminar for the semester because he can give us a really interesting perspective on both being in academia and industry. So Adam and his brother Ryan founded Embark, a dog genomics company. You'll probably can correct me later, but it's sort of like 23andMe, but for dogs. And Adam's the chief scientific officer. And what's really cool is that they use some of this DNA with the owner's permission for their research. Adam, we're really excited to hear your seminar. Thanks for talking for us today.
Thanks. Thanks. I'm really happy to be here virtually, or however we want to say that it is. I will say the coolest thing about starting Embark was that if you guys tuned into Jeopardy on April 1st, we were the question of Embark, which is kind of like 23andMe does this for dogs, and the person got it right. So Embark was on Jeopardy, so I think we can all close up shop now and be happy. But anyway, I'm very happy to be here. My background is in biology and computer science. I've always been interested in understanding the evolution of biological diversity. My PhD work was in heliconius butterflies and trying to understand how you evolve novel warning patterns. And I started my postdoc, working with human genomic data sets and looking at the structure of genetic variation in people and the amount of it that's due to neutral versus deleterious mutations. And I quickly transitioned into dogs sort of by chance. My PI, Carlos Bustamante, had a collaboration going with some eminent canine researchers at NIH and UCLA. Looking at the diversity of dogs and the genetics that underlie it. And dogs are a really great model. They've got, of course, the plant world is full of tons of diversity, but in the mammal world, dogs have the largest range of variation in body size of any species. And they also vary in tons of other ways. And because we've selectively bred dogs to have different behaviors and traits and fill different roles, it leads to, in many cases, extreme behaviors being caused by just one or a handful of mutations. So for instance, achondrodysplasia, which is limb dwarfism. So you see that in beagles and corgis and stuff. That's the same mutation in all of those breeds. It's just one mutation. Brachycephaly, there's a few mutations that cause it, but it's usually in any breed, there's one mutation that leads to the short snout phenotype. And even things like body size, where there clearly are several genes that are doing it, the genetic underpinnings of it is a lot simpler than what we would see in, let's say, humans or most randomly breeding populations. And so, one of the first projects my lab did here at Cornell was genotyping dogs for, doing an expanded CAN map where we looked at 158 different breeds. And so if you genotype about 10 dogs from each of these, so this is less than 2,000 samples that we looked at, you very quickly can identify 18 different genes that are associated with body size, p-values. For those of you who are unfamiliar, this is a Manhattan plot. So this is how geneticists do association studies. And the goal here is these are the log of the p-values. So this is kind of like the skyline of Manhattan, and the taller the skyscraper, the more significant that locus is. And so we see all of these genes that, make sense, insulin growth factor 1 being the largest one here, for for causing the body size differences that we see in dogs. And if you were to do a comparative study in people, GWAS study in people, the p-values are much lower. It takes much larger sample sizes. So it wasn't until we had like 1,000 people that we had, heights for and genetic data for, that we started to see the first genes. And some of these genes are the same, like HMGA2. And as a consequence, we can actually predict from the genetics the adult expected body size of a dog very accurately. We can explain easily 85% of the variation just looking at 24 genes. Whereas in people, you'd have to look at hundreds and hundreds of genes, and you're only going to explain about 20 to 30% of the variation, from that. So I wasn't just interested, of course, in trait mapping. So, my background is how natural populations evolve. And a neat thing about dogs, actually, is that most of the dogs in the world are not purebred dogs, even though most of the research in dogs is done on purebred dogs. And in fact, most of the dogs in the world aren't even mixed breed dogs, which descend from purebred dogs, but then have been, crossing with each other most of the time of their own accord. But we also have designer breeds where it's sort of deliberately done. Most of the dogs in the world are what we would call free-ranging, free-breeding dogs or village dogs. And so there's about 1 billion dogs in the world, and easily 60 or 70% of them fall into this category. And this is kind of how dogs initially evolved, right? So dogs descended from gray wolves, and when they descended from gray wolves, they didn't turn into poodles. They were village dogs. So basically, the lead theory is that they domesticated themselves. So wolves and human hunting communities were going after the same large prey. And if there were climatic changes or humans got better at hunting or otherwise there were hunting pressures on the wolves, it started, some wolf populations would do better if they could scavenge from the humans. And in order to do that, you need to tolerate the presence of humans. You need to evolve this sort of tameness, this ability to read human cues, so you know whether or not the human is a threat to you. And then other things that go along with scavenging, like smaller body size, so you're less threatening and you don't need as much provisioning, and being able to reproduce multiple times a year. And so eventually, once you start to go down that path, you're not going to go back to being an obligate hunter of large prey anymore. And so it sort of set the stage for this one small group of wolves to expand out into dogs. And so now we have 1 million wolves in the world and 1 billion dogs in the world. So it was a highly successful strategy. And so, yeah, most of the dogs in the world are still indigenous village dogs, although once the tameness had evolved, you could start breeding these dogs. You could start selectively provisioning or selectively mating certain dogs. And this is how breeding typically had been done. So thousands of years ago, we had sled dogs, we had sight hounds, we had Molosser type, guardian dogs. And then much more recently, especially during the Victorian era, we started these closed purebred registry populations. And that really changes the dynamics of the dogs. It sort of accelerates drift and selection in those lines, makes them clearly genetically delineated, but it's very different than how traditionally breeding has been done, where you're trying to achieve a certain working ability generally, and you're willing to cross in and out in order to achieve that. The consequence on the genome is predictable. So if you look at a Labrador and you do a whole genome sequence in the Labrador, you're going to see there are many genomic segments that are identical by descent, some of them quite large. This is not an atypical lab. Most labs are have 20% of their genome inbred or so. This lab has all of chromosome 23 inbred. And if you compare that to a village dog, the IBD sexes are much, much smaller. So, even though superficially looking at them, you might not always be able to tell, is that a village dog? Is that, what? But genetically, they're very different. And this gives you information that you don't get from purebred dogs, right? So one of the questions about dogs is, where do they come from? And, people have looked at many different areas, and there were theories that, they came from East Asia, they came from the Middle East, they came from Europe. And a lot of these studies have been biased because the genetic analysis was done primarily in purebred dogs. And, like trying to understand human origins by looking at European royal families, you're not really going to capture the full amount of genetic diversity. Only some of the diversity that's in the natural dog populations actually made it into these various purebred lines. And so, in fact, by going around for the Village Dog Project and sampling these dogs, we could look at patterns like linkage disequilibrium and see what the rate of decay was in the different populations. And we clearly got a very different picture than what other groups had been seeing, where it looks like Central Asia has this clear signature of a center of diversity. And so we think that makes the most sense as a origin for domestic dogs. In my lab, we also have tried mapping other traits of interest. So one of the things we looked at was how much each breed of dog sheds. And in fact, if you can characterize that, you can actually do a genetic mapping project. And we found a new locus that contributes to shedding behavior. So we've always known, we've known since the CanMap project of this R-spondin locus on chromosome 13 that gives kind of the wiry hair-like coat for poodles. That's low shedding. But there's the secondary locus, this melanocortin receptor on chromosome 1. And so for dogs that don't have the poodle coat, if you've got the wolf variant of this MC5R allele, you're going to be high shedding. Whereas if you have the boxer variant, you're going to be low shedding. And so that's also a trait that, a lot of people might be interested in. We not only looked at things that we think have been under selection in dogs, but because my lab was generating a lot of dog DNA data, so these studies here are based on 4,000 dogs, so that was the largest genetic analysis of dogs at the time. We could go back, and most of the dogs that weren't village dogs that got collected were collected at the biobank at the Cornell Vet School, and so they had veterinary records attached to them. And so one of the things many dogs that go into the Cornell Vet School get are blood panels, right? CBCs and blood chemistry panels. And so we could go back and we could look at the data from those and see if anything associated with the genetics of the dog. And one of the clear associations we got, so this is a zoom in on a Manhattan plot at the locus on chromosome 13, where we found this significant association of ALT, so alanine aminotransferase. It's a signal of liver damage. And so if you have high values of it, it means that your liver is not functioning the way that it should. And it's centered right over this gene GPT, which actually encodes the transferase. So it makes a lot of sense that if there is going to be an association, it's going to be there. So there's some variant here likely associated with this GPT gene that leads to very different values of ALT when healthy dogs are tested. And so the reference range, it's published that veterinarians use. It's somewhere around 12 to 99 is considered healthy. And if you look, the average values in healthy dogs generally fall within that, but they're different depending on whether it's the wild type GG or whether it's homozygous for this AA mutation and heterozygous is intermediate for it. And the reason that's relevant is if the dog actually does have liver disease. So then we went back to dogs that have been diagnosed with liver disease. And then you look and you see what the ALT values are. Well, sure enough, they're all elevated as you would expect. But the dogs that are AA don't elevate nearly as much as the other dogs do. And in fact, half of them or more are still in the normal range, even though the mean is up to 181 because it's on a log scale. So you have some dogs that really drive that. So you're going to miss the diagnosis if you're relying on this and you don't understand the interaction with genetics. So it's one of these things where reference ranges are published for species in veterinary world, or at best for populations, but really they should be considered on an individual level. And if we can get the genetic data that we can more accurately predict what an individual's healthy range is, we can probably do much better in the clinic. So we've looked at patterns of selection across all the dog populations in my lab. And we can see that there's many regions of the genome that we can associate now with selection for body size, body shape, skull morphology. There's other regions of the genome that we associate with fur traits, such as shedding or wire coat or coat color. We see signals of positive selection on the dog MHC region, the dog leukocyte antigen area in chromosome 12. But we also see lots of regions of the genome that have signatures of selection that we have not associated with a phenotype yet. And so this has led to, we have low-hanging fruit of the dog genome, and we just have to ask the right questions to figure out, what's driving selection, which lines is this being driven, and how do these selected traits evolve? So, I would say that dog geneticists have made a lot of progress understanding what we would call Mendelian disease. There's over 250 diseases that we know the causal mutation for in dogs. So other than mice and human, I think we know more about Mendelian disease in dogs than any other species. We certainly know a lot about the genes that control major morphological traits, but we don't know as much about the genetics of complex disease. We don't know the genes that go into hip dysplasia or most of the genes involved in cancer, or being able to predict the risk and being able to, breed dogs that are less at risk using genetic testing. We haven't found too many genes involved in behavior. I mean, the one exception that sort of proves the rule is there is a locus for canine compulsive disorder, and it actually falls in the same gene that's been associated with human obsessive compulsive disorder. So we think it's the same gene. We just can't talk to the dogs to know if they're obsessing or not. So we call it something else. But for other behaviors that we've sort of positively selected for, like prey drive and things like that, we haven't yet made the associations. So it's more complicated there to do it. And we probably don't have the right sample sizes that we need to do this. And we've also made slower progress understanding how genetics and longevity are related. We know that large dogs die sooner, but beyond that, we don't know exactly why. We don't know if it's specific genes that are causing this, if there's ways to breed big dogs to live longer using other genes. Why do cats live longer than dogs? Like there's lots of stuff that we don't understand, and dogs, because they have such a range of size variation, and because that's associated with a twofold variation in expected longevity, they're a really nice model for trying to understand how genetics and healthy aging interact. And so basically, the limiting factor here is that large-scale studies are needed, right? So the 4,000 dogs that my lab looked at are not going to be enough to tackle these questions. We know from human genetics that it's going to take many, many more samples, probably hundreds of thousands, in order to do something like this. So I was at the stage of my research where, okay, well, we've now looked at the genetics of nearly 5,000 samples that we've got in a biobank. We've made some cool discoveries. The biobank has 20,000 samples. So, if we could get funding to look at more of them, that would be great. I've already kind of tapped out NIH funding. We got National Geographic funding. Like, where do I go to next to do this kind of big data thing? How am I ever going to get hundreds of thousands of samples so that I can study cancer and I can study behavior and we can make progress on allergies and all sorts of stuff that, people are really interested in. So at the same time I'm thinking these things, of course, we've been recruiting dogs in the community to be involved in studies. We've got some, press buzz from that. We get lots of people asking, can my dog take part in your research studies? And can you tell me, what my dog is and why he is the way that he is? And so it sort of, got this idea that if we could do genetic testing the right way, we could help people understand their dogs better, give them the information that they would need to, to understand what lies down the road, what risk factors, should this dog be bred, who should it be bred to, things like that. And then we would get all of this data back that we could use to actually build really large research studies if something like this was popular, if there was enough demand for it. And so the idea is personalized genetics for dogs. So we would want to, for the same things that you do for humans in 23andMe, for instance, we want to predict disease predispositions. We want to identify relatives and ancestry. People want to participate in citizen science research and some sort of explanation of, why do I have red hair or why do I sneeze when I walk out of a movie theater or things like that. And so, and the neat thing is there's 4 million Americans born every year, and there's 8 million dogs that are born in America every year. So there's this huge potential for research study if this takes off. And so that was the idea for starting Embark. And so my brother, who had been participating in the Village Dog Project, traveling around the world off and on over the course of a decade, as he was graduating from Yale, we talked it over, and so he decided, yeah, we'll take the plunge. And so he's CEO, and I'm the science guy for the company there. And so we so the idea is we use the same sorts of Illumina genotyping arrays that researchers use, although we add in specific markers for specific analyses, but ideally it's this discovery platform where you're looking at over 200,000 markers. And so it gives you a lot more data than any other test on the market, which makes it, more accurate for doing lots of different stuff. And if we can keep customers engaged, it will fuel discovery. And so over 90% of the people who do Embark have their dogs opted in for participating in research. Most of them have filled out surveys about their dogs. So we have this information about the dog's health, their temperament, or environment. And then we can update as new discoveries are made, whether the dog is predisposed for something or something like that. So, my dog at home is Penny. So Penny, we got from a shelter up in Rochester. She was labeled a Basenji Terrier mix, which I thought was odd. And sure enough, when we actually genetically analyzed Penny, we saw that, well, she was a terrier mix, but she had a lot of other stuff that wasn't Basenji that explained why she had a curly tail and all that other stuff. And so, with Embark, you've got research, health, breed, traits, and relatives as possible, areas that you get results for. So for the health, Penny's clear of the genetic conditions that we tested for at the time we did the test. We've expanded the chip now. We've got more and more stuff. You get a family tree, which is sort of an estimation of, how we think the breeds got into her. Of course, you don't have a crystal ball. You can't always get the family tree 100% accurate, but you at least know, like, is the Jack Russell Terrier on both halves of the family tree or just one half, and things like that. We can look at the various coat color genes and other, traits that we can map. And so we can look specifically at the E locus, which variant does she carry, at the B locus, what variant does she carry, at the D locus, what variant does she carry. And we can combine all of these, to help the user see what should the coat pattern of your dog look like. And so in Penny's case, she's tip sable. And so sure enough, yeah, so she's got the white underbelly and chest and the brown back and black on the tail and ears and snout. And then what I thought was really cool is we got to see relatives. And so, after about a year or two, we actually had a cousin of Penny that wound up testing with us. And we had no idea that these dogs were related. These dogs were adopted a year apart. But Dharma, Penny's cousin, also lives in the Ithaca area. And so we've actually been able to go out and visit Dharma. And it's actually pretty incredible how similar they are. It's like the same chassis, but different paint job. And a lot of the behaviors are very, very similar between Penny and Dharma. And they have some health issues, like they both have had lots of teeth removed for enamel hypoplasia. So we think there's a genetic component to that. And so, most people don't know their dog's family medical history. So it is kind of cool when you start seeing that there's relatives of your dog and reaching out to them. Penny is not inbred, she's outbred, and we're not breeding her. So it's less of an issue, but we also can look at dogs. So this is I believe a Doberman Pinscher, that we looked at, and we can calculate the inbreeding, not from the pedigree of the Doberman, which only goes back a certain number of generations, and it's just an estimate anyway. We actually look across the genome like we did for those whole genome sequence dogs at the beginning of the lecture, and we can see which parts of the genome are identical by descent and total that up to get an inbreeding coefficient. And so that's another thing that you get to learn about your dog. And then, finally, to close the loop, you can also take these surveys and assessments for your dog. And so we're asking customers things about, does your dog have allergies? Are they food allergies or seasonal allergies or, things like that? And with the idea that we're building this database that's going to drive discovery in the future and make DNA testing even more valuable in dogs, and hopefully provide avenues for therapeutics, for reducing the prevalence in breed dogs, and for translational research where you have diseases that are common in dogs and easier to study in dogs than they are in people. So yeah, the comparison to 23andMe has been made several times. So 23andMe came around about 9 years before Embark started. They launched their first genetic test in 2007. And just about 3 years later, they got their first scientific publication. So again, this is humans were testing themselves to learn more about their ancestry, to learn more about their health predispositions. And then they were filling out very detailed surveys about themselves so the scientists could see if they could make any more associations. And so the first paper that 23andMe, the first associations they made were freckling. So this is the BNC2 gene. So it wasn't known to be involved in pigmentation at the time. But it was clear that it explained a certain fraction of freckling in the customer database. Photic sneeze reflex. So this means if you are in a dark room and then you walk out into bright sunlight, do you have this urge to sneeze? And so this ZEB2 gene is interesting because it's actually been implicated now for light-induced epilepsy. So we think that the mutation that's driving this association is just a subclinical kind of epileptic type thing maybe going on. So it's kind of an interesting model now that we wouldn't have known about without that sort of testing. And then the third trait that they published was asparagus insomnia. So this is whether or not after you eat asparagus, you report that your urine smells. So again, these were very detailed surveys that people were filling out. And as it turns out, this trait maps significantly to an olfactory receptor. So a lot of scientists thought it was a metabolic thing. Some people broke down asparagus different ways than other people did, but it's actually, it's an olfactory receptor. So everybody breaks down asparagus the same way. It's just some people smell the metabolites and some people don't, and people don't typically smell other people's pee. So that's why we had that association. So these seem like kind of, fun, but not really like important for health sorts of traits. And that's maybe true, but down the line, they definitely did, make significant contributions as their database increased in size, and they could ask, more and more health-related questions, and they've identified novel risk factors for things like asthma. They did a lot with Parkinson's research, osteoporosis, tons of major medical conditions that honestly, it would have been difficult, if not impossible, to get the same sample sizes. It certainly would have cost a lot of government funding or something in order to do that. And so it really contributed to research, as well as enabled research that just wouldn't have been done otherwise, almost certainly. So when you have large data sets, you can ask all sorts of questions and you can get really cool findings about there's genetic associations for educational attainment, age of reproduction, mosquito attractiveness and symptoms, things like that. So that they aren't really medical, but they're just, if you're interested in that kind of stuff, they are, really interesting things to look at. So with EMBARK, the first trait and the first publication we get was a little bit of a surprise. It was just one of those questions that we asked and we saw this, clear association signal for, and it was blue eyes. So what color are your dog's eyes? That's something people can answer really easily. We run a GWAS analysis of that. We saw the association that we expected on chromosome 10 at the Merle locus. So this is the funky coat that some Australian Shepherds and other dogs can have, and that can lead to blue eyes. But we saw an even stronger association on chromosome 18, which was not previously identified as having to do with pigmentation at all. And it was driving the blue eye phenotype that you see in some Siberian Huskies and related breeds. And sure enough, we looked at the array data. We could see that there is a conserved haplotype in many of the Siberian Huskies that have it. Although we saw this sub-region that all of the dogs that were homozygous had a heterozygous gene call. So that's not expected. And so that's an indication that there's likely a structural variant going on there, which is making the genotyping weird. And so sure enough, when we look at whole genome sequence data that's been publicly deposited, we can see that Siberian Huskies show a duplication across the region spanning those markers. We could actually do a breakpoint analysis and identify, specifically where this duplication, it's a tandem duplication. It's about 99 kilobases long. And we could ask, okay, so was it the SNP association or is it really this duplication that's driving the phenotype? And so what's great is we have the contact information for everybody that tested with EMBARK. And so we said, okay, well, that was our discovery panel, was everybody who answered that survey on the website. Now everybody who didn't answer the survey on the website, let's email them and ask them what color their dog's eyes are. And so we have this validation panel that's in place. And so we could get another 2,700 dogs, just in the matter of a weekend, filling this out. And then we can look and we can say, oh yeah, so that SNP is still associated in our validation set, clearly 10 to the minus 120th power, but the duplication is much, much more associated with it. And so that's likely what's driving the phenotype. And I think this is probably the most significant p-value in canine genetics so far. And it was the biggest mapping study in dog genetics, so 6,000, dogs. So if I haven't driven home the point of big data being important, it is. And so looking at the things that we want to look at, we want big data. And fortunately, the database has been growing. So if we look at the number of dogs that we've got genome data for where the owners have consented for scientific research, we see that it keeps growing year after year, and we're up to about 250,000, right now. And so the kinds of studies we're able to start undertaking, are a lot more useful. And the reason why it's growing is because, we've got, high customer satisfaction. So they like what they're seeing. So I think we're 4.7 stars on Amazon or something like that, which is not only higher than other dog DNA tests, but it's also higher than I think any other genetic test that's out there. And it's mostly been driven by people wanting to know what mix their dog is. So most of the customers are mixed breed dog owners, but we do have a lot of purebred dogs and breeders, and they know what breed their dog is, but they're doing it for the health testing or relative finding or inbreeding analysis or stuff like that. And then that tiny little sliver there is primarily village dogs. And so, one of the ideas of starting the company is, man, we could probably get a lot more data if people just sent us their dog's DNA than if we had to travel out into the remote rainforest to try to get dogs. And so we only have a sliver of village dogs that are going in there, but that's still over 3,000 dogs. So it's actually, 5 times more than any other village dog analysis that we've, been able to do. And many of these dogs are coming from places that would not have been feasible, or cheap for us to go to and to sample. So that's been really nice too. So to put it in perspective, it took my lab, basically 5 years to get the funding and to do the analysis to get, 5,000 dogs analyzed. And on Cyber Monday, this last year, in one day, we had 10,000 kits that were sold. So we got twice as much data. So it's a very effective way to get, large amounts of data. What's interesting is although there's high customer satisfaction, you do have the occasional customer that's very, very unsatisfied, and to have to walk through the genetics, so we had one customer whose dog came back as half golden retriever and half soft-coated Wheaten. And they're like, well, this is impossible. My dog is black. So there's no way that a gold dog and an apricot dog are going to cross to make a black dog. But the great thing is we're already testing all the health and trait things. And so you can walk through the genetics and you can be like, oh, well, actually golden retrievers are golden, because they're recessive at the E locus. And soft-coated Wheatens are gold. Are apricot because they're recessive, they've got recessive yellow, but that's the K locus. And so then when you cross them, you get 2 heterozygotes. And so it's not surprising that you have a black dog. And so you have to do a lot of customer service in order to sort of explain these things. Here's a customer that was complaining because they knew their dog's results had to be wrong. Their dog clearly was a breed, that was not in there. And you look at the results and you're like, well, I mean, I don't know, it looks reasonable. What breed is supposed to be in there? Whippet. I'm like, oh, a parent was a Whippet or something? That would be, it should have come up on the trait. How do you know the dog is a Whippet? Well, I don't know who the parents are, but if you look at the dog, it's got the Whippet smile. It has to be part Whippet. And so, a lot of stuff is, you have to walk people through and get them to understand it. And some people really have unreasonable expectations. So they go to a shelter, they adopt a dog that looks like a border collie. Of course, the shelter tells them it's a border collie, but genetically it's not a border collie, just superficially resembles one. And so you have lots of, lots of hand-holding that has to go on. But overall, people are really pleased to actually know what's in their dog, because it helps them kind of understand where their dog's coming from more, maybe encourages them to do different stuff. From a vet perspective, though, one of the neat things and one of the projects I've been working on most recently is the question of, is genetic testing actually worth it for mixed breed dogs? So all these people that just want to know what breeds are in their dog, is it actually worth it? Can we test for stuff that's relevant to enough dogs to really justify the idea of you should do genetic testing for that? And of course, obviously, conflict of interest, I have a dog DNA testing company, so I'm going to be biased, right? But if we actually look at the numbers here, so we've got several genetic conditions that are at reasonably high frequency. So none of these are common, right? These are all bad diseases. So they're not going to be at high frequency, but they're still, at 1% frequency, or higher. So these are the allele frequencies underlying. So PRA is a form of blindness, and there's a couple different kinds of PRA that are in dogs, and they're generally in certain breeds of dogs and not in other breeds of dogs. HUU is hyperuricosia, so it's bladder stones. So if you've if you're predisposed to that, all Dalmatians are predisposed to that. If you've got that predisposition, you have to be fed a special diet. Otherwise, you get these really painful stones, and it can take a while to diagnose sometimes, and you know, it costs a lot of money. CEA is another form of blindness, common in collies and related breeds. EIC is exercise-induced collapse. Ichthyosis is a skin disorder. So all these things are recessive traits. And so if we look, they're at a certain frequency in mixed breed dogs. Generally, they're at a slightly lower frequency in purebred dogs because breeders have tests and they know what lines carry which, and so they can try to reduce the frequency within the breeds in a way that you know, mixed breed dog breeding doesn't really have that ability unless you're testing for everything and actually trying to breed healthy mixes, which generally isn't how mixed breed dogs are made. And then village dogs have the lowest frequency. So these are all things that are under selection, right? They're common in a lot of breeds, so they are ancient mutations that are in a lot of lines, but they've gotten in much lower frequency due to natural selection in village dogs that isn't there for purebreds and mixed breed dogs. Even though the mixed breed dogs tend to carry a slightly higher frequency than the purebreds do, they're less at risk because they're more outbred in general than purebred dogs. You can have inbred mixed breed dogs, but on average, they're more outbred. And so overall, the at-risk frequency, if you've got a mixed breed dog at home, for these tends to be quite low. In fact, if you sum up all of these, and so I'm going to say there's one more disorder that we test for, which is SOD1. So this is degenerative myopathy, which is like canine ALS, and this is also recessive, but it's late onset and it's very incompletely penetrant. So it seems like it's only predictive in certain breeds and not in other breeds, and so it's not really clear that that's an actionable test right now, at least for mixed breed dog owners. So we're going to ignore that test for now, and we're going to say, well, just looking at the actionable recessive disease test that you can do, only about 1% of mixed breed dogs are actually at risk for those. So only 1% of dogs, if you're not planning on breeding them, is this going to be useful information. But in fact, there's also MDR1, so multi-drug resistance. And so this is a drug sensitivity, not just to like one drug. It's like whole classes of drugs, chocolate, like different kind of food things. You know, it's the body's ability to kind of cleanse different toxins out. And so this is a very relevant, it's not really a disease, and it's additive. So even if you have one copy, that leads to some sensitivity, and you want to know about that. And if you have two copies, it leads to lots of sensitivity. And so just adding that trait in there, suddenly we've got 3% of mixed breed dogs that are getting an actionable result. And then recently there was this publication about intervertebral disc disease in dogs. And so this tends to happen to like long dogs, low riders. And there's actually a mutation separate from the chromosome 18 mutation, which gives you that phenotype, but without predisposing for IVDD. There's a chromosome 12 mutation. So it's a retrogene. It's the same retrogene that causes the short limbs in basset hounds and dachshunds, but it's in a different chromosomal location, and it leads to brittle discs. And so much increased likelihood of suffering from IVDD. And so they estimated that 3.5% of all dogs get IVDD, and it's essentially only dogs that carry 1 or 2 copies of this. So there's a 50-fold risk increase if your dog has 1 or more copies of this disease. And so suddenly, if that holds true for mixed breed dogs, so they only looked at purebred dogs, but if that holds true for mixed breed dogs, then suddenly you've got 17% of mixed breed dogs, because again, this isn't a recessive disorder, this is a dominant disorder, are going to get an actionable result. If your dog is at risk for this, you don't want them jumping up, you want to get ramps for the stairs, you want to give them different supplements, because it's a very expensive and painful disease to treat. So we, of course, designed our array, designed a test so that we could see whether or not a dog carried this variant, and we also could survey owners. And so we could ask the owners, you know, of all, we asked them several questions, and one of them, IVDD was one of the diseases we asked about. And then we also asked whether they did any changes to their dog's routine after they got their genetic results. So even if their dog hasn't been diagnosed with IVDD, if they see that it's at risk, is there, you know, did they do any of these things that would help mitigate that risk? And so we could, you know, very quickly turn around a survey with 782 respondents, and we could calculate that dogs that had 1 or 2 copies of this disorder were 43 times more likely to have gotten diagnosed with it, and over half the dogs that hadn't been diagnosed with it had changed their care routine. And so, of course, as we follow these dogs through time, it'll be interesting to see whether the dogs that changed their care routine before a diagnosis were more successful at staving off the diagnosis or not, or which interventions, made sense. But it definitely seems like now, about a sixth of all mixed breed dogs can get an actionable result. And of course, this keeps growing as more and more discoveries are made. So the other area that my lab and Embark has been interested in has been inbreeding, because we see very large differences in average inbreeding when we look at different breeds of dogs. So Dobermans are very inbred, Beagles are much more outbred. When we look at data for mixed breed dogs and purebred dogs from other published, from other sources like veterinary records that don't have genetic data associated with them, we see that, of course, there's a body size effect. Big dogs don't live as long, but for a given body size, mixed breed dogs seem to live a bit longer than purebred dogs do by about a year or more. So now that we have a large amount of genetic data, of course, most of the Embark dogs are young. People tend to get the test right after their dog. They got their dog or they heard about Embark. But we have had, fortunately or unfortunately, depending if you're a scientist or somebody who loves dogs, a fair number of dogs that have passed away. And so we can look at that longevity data and ask ourselves whether or not it seems to be associated with inbreeding. And so in fact, we've got about 1,000 dogs now with complete longevity data. And we can see that, whether the dog is spayed or neutered has an effect. Obviously, weight has an effect, but coefficient of inbreeding also has an effect. And so, you lose, easily, 2 years of life if you go from 0% inbreeding to 50% inbreeding. And in fact, that pattern holds both for purebred dogs and mixed breed dogs. So it looks like the age curves are basically identical for the two, and it's just most mixed breed dogs are, over here completely outbred, and only a few of them are inbred, whereas with purebred dogs it's much more evenly distributed. And so if we look within a breed, so we've had a lot of Dobermans that have been tested. We have, clearly the Dobermans that were the most inbred had shorter lifespans than the Dobermans that were the most outbred. And again, if we look at even larger data sets of pedigree data, we can see that the most inbred golden retrievers have shorter lifespans than the most outbred golden retrievers. So there seems to be a longevity effect for inbreeding. And beyond that, we actually collaborated with Morris Animal Foundations that's running a golden retriever lifetime study. And we did a genetic analysis of about 100 of their female golden retrievers that had been bred. And we could see, even with that small sample size, there's clearly this effect that the more inbred females had smaller litter sizes. So there's lots of evidence to suggest that we've got inbreeding depression in dog populations today. And in fact, so we've got 20% of purebred dogs can be at risk for something, and so it's important to do genetic testing for that. But it's also the case that if you were to do your breeding so that you move more of the dogs from the high inbred side to the lower inbred side, you're also going to improve the health of dogs by at least that order of magnitude. And so for breeders, we now have a matchmaker tool where you can go through and you can see dogs of the opposite sex, and you can see what would the expected percent inbreeding be if that dog was bred to your dog, and you can see where it falls on the on the line, and hopefully you can try to prefer more outcross dogs and more than more line bred dogs through that sort of thing. So that's pretty much what I came here for. Obviously, we're doing more genetic research into health conditions now that we have this really big database. We are recruiting for certain studies like dilated cardiomyopathy, tricuspid valve dysplasia, wobbler disease. So we are trying to now leverage this big data set to make the health discoveries and go from there. So hopefully next year when we give this talk, we'll have some really cool stuff like that. Right now, we've got a lot of cool trait analyses and associations and stuff. But yeah, so in conclusion, we do see recessive disease in mixed breed dogs. And we see Mendelian disease with IVDD. And so genetic testing for health actually seems like it makes sense for a lot of mixed breed dogs. But beyond that, of course, testing for these has always been valuable for purebred dog breeders. But actually looking at COI is going to be just as valuable. And so getting the tools out there so that they can use genetics to breed outbred dogs is going to help really make dogs healthier. And then, beyond that, just if you can do the genetic testing in a way that is done on a research type platform, a dense genotyping or sequencing platform, so that data can then be useful to drive association and discovery. There's lots of opportunities, for genetic improvement of dogs and for getting new genetic models for traits and diseases. So with that, I'd like to thank you and thank everybody that contributed to this. This is just a small number of people whose works I happen to cite in this presentation. And I want to remind everybody, it is DNA Day on April 25th. And so for the entire month of April, they're running a $50 off promo. So if you wanted to do your dog, now is a good time to do it. So with that, I'd like to open it up if anybody has questions. I'm not sure how to handle questions on Zoom, so if I don't do it right, I apologize.
So I think we can have people, if you want to ask a question, go ahead and raise your hand on Zoom, or you can just unmute yourself and start talking. Okay, well, so I have a question. How many people work at Embark, and what?
About half of them. No, I'm kidding. We're up to Embark. So Embark has offices in Boston and in Ithaca. So the science team is in Ithaca, and it comprises about 10 people. And then the Boston office has about 50 people.
And what kinds of people do you hire to work there?
Oh yeah, that's a good question. So a lot of it is, software engineering to maintain these large databases and to efficiently deliver results and reach out to relatives. And like if you have a relative database and you've got 250,000 dogs, that means you have 250,000 times 250,000 relationships, right? So there's lots of interesting big data problems, and we also do methods like imputation. So sure, we have direct genotypes of 220,000 markers, but actually we impute millions of markers so that we can do the discovery studies and stuff. But we, obviously we have a logistics team, we have a product team, so there's, lots of, and, things like HR and finance and boring stuff, right? And then scientists, and we like people with both laboratory skills. We do our own, even though we outsource the commercial laboratory stuff to like a CLIA certified lab that handles, millions of samples and, has the ability to scale up. We still maintain a research wet lab here in Ithaca. So there's lots of, different skills that we look for, not just on the computational side.
Okay, other questions?
And veterinarians, we hire veterinarians.
Okay.
Yep.
You can go. Oh, okay.
Go ahead, Francis.
Hi. I'm just wondering, how do you get the genotypes of the village dogs? So how do you get the DNA? Do you have someone going around the world and like? Yeah,
so I should have clarified. Yeah, so we actually started that project. We had Cornell seed money to go. And so this was 2007. And so my brother on his honeymoon to Africa started sampling village dogs in Africa. And we actually analyzed 350 of the dogs that he sampled on his honeymoon. And it was a pretty cool analysis. Nobody looked at village dogs in Africa before. We got a PNAS paper out of it. And then we got NSF funding to expand that to a more worldwide thing. And so we worked with local veterinarians. Ryan or I traveled to several of the places. We were taking blood draws from the dogs. And so, and we would, because of shipping issues, a lot of times you'd have to like spin down the blood in place. So Ryan had to like practice how to explain centrifuge in Swahili at some of the border crossings. But we did that analysis, and then that continued when I started on the faculty at Cornell, and we had collaborators in Mongolia, and I soon go to Nepal, and all of these sorts of things to get some of the key samples for that analysis. And now, for the most part, it's customers sending us samples and letting us know where they got their dog from, and we get GPS coordinates and all that kind of stuff.
Interesting.
Yeah.
Cool.
Yeah, it's amazing where some people have gotten their dogs from. And it's also amazing the reaction of some people, right? Like, so somebody's like, well, you know, like you said, you showed a map of my dog, and that, you know, you said that my dog was an Indian village dog, but I wanted to know what breeds were in it. It's like, well, you know, like dogs have been in India for like millennia, like way before any of these breeds came along, right? That's what it is. It's not a mixed breed dog. And then other people are like, wow, you had a map and it was like right on Delhi, and that's where I got my dog. It's like, well, that happened to be a city that we sampled, so we were able to get it right.
Cool.
What was the transition from being an academic scientist to being like a chief scientific officer of a company? What was that like?
Yeah, it was pretty crazy. So, you know, I was running my lab, and I'm still running my lab, but you know, so starting the company was, I mean, the impetus for starting it was that we had gotten funding actually from a private company to design a dog DNA test. But it was through Cornell, and so it's Cornell IP, and we got all built up, and then that company is like, well, it's not really in our wheelhouse. This isn't. We've decided we're going to go in a different direction. So you guys do whatever you want to with it. They're like, all right, great. And so then we talked to another company. Hey, do you want to use this dog DNA test? We think dog DNA testing could be a lot better. And we went to several different companies that we thought would be interested in doing this, but nobody wanted to kind of take the leap to do it. And so I was kind of like stuck. But then when my brother was graduating, he's like, well, you know, I need you to help me, Adam, because I'm about to graduate with a PhD, and I don't want to be an academic, because everybody I know who's an academic lives a miserable life. I'm like, Ryan, it's not about you, it's about me. And so we talked about it, and then we started to say, well, maybe there's a business model idea here. Maybe instead of trying to like get another company to do this test to get the research data, we just start a company, and you run the company, and, you know, I'll provide the scientific input. And so we went to the Cornell Technology Licensing Office, and we sat down, and, you know, it's a little bit different of a licensing than they generally do, but they were on board. It's been great. We actually get to help Cornell in various ways, and they certainly have been a big help, you know, getting us started and going. And then so then I went on sabbatical after I got tenure. So for the last year, I've been working for the company, and now this is like my first week back. So, you know, so I'm back now.
Cool. I have another question, which is, how much does what you're learning in dogs about diseases translate over to humans? And are we, you know, is it helping us understand human health even better?
Oh yeah, absolutely. I mean, there's a lot of great stories about translational research that's been done. So there's, you know, one of the first Mendelian diseases in dogs that was identified was narcolepsy in Doberman pinschers, and the gene that's defective there wasn't a gene anybody realized was involved in sleep cycle signaling. And so in fact, after the dog geneticist figured it out, the human geneticist went and took a look, and it was like 25% of the cases of narcolepsy that they had no explanation for it were due to defects in that gene. Another instance, Duchenne's muscular dystrophy. So dogs also have a defect in the same gene. And so there's a colony of dogs that they raise so that they can do Duchenne's muscular dystrophy studies. And so they testing different drugs and therapeutics and stuff like that. But genetically, one of the interesting things to come out of that was that in that colony, a dog tested for it should have the disease, but didn't have the severe phenotype at all. It was so mild it wasn't even noticeable. Like, dogs seemed normal. And they thought that was odd. They thought there must have been something in the upbringing of this dog, or, you know, it's really kind of an outlier. We don't know what's going on. And they just kind of chalked it up to randomness. But fortunately, the colony wasn't run that well, and the dog escaped and wound up siring 3 litters. And several of the dog's pups also tested at risk, but didn't have the Duchenne's phenotype. And so then they started doing genetic testing. And so then they found this compensatory mutation in a completely different chromosome that led to upregulation of this jagged gene. And so that seems to be what's protecting them from the severe phenotype. So now you've got this new drug target that you can start to look at. to see if you can design something that will upregulate, and is that going to help in the human case. So there's a lot of the genetic, most of the diseases in dogs have human equivalents, and many cases it's the same gene. In some cases, it's the same amino acid that's being mutated that's leading to the disease. So there's lots of opportunities for translational medicine that way. What we'd like to do is, I think dogs are a great model for aging research, for cancer research, but we just need to know more about the genetics of it to really accelerate that kind of translational research.
Yeah, sounds like you're getting there. It's going to be really exciting.
Yeah, definitely.
Any
the dog
other...
genome got published in 2005, so that was the first year I started my post. That's the year I graduated with my PhD, so it was sort of the right time to get into the field.
Any other questions, Francis?
So you said that there's a gene in dogs that's associated with compulsive behavior that's also involved in obsessive compulsive disorder in humans.
Yep.
I'm wondering what gene is that, or like how strongly is it correlated with obsessive compulsive disorder in humans? I'm
not familiar enough with the human literature to go out on a limb and say, obviously it's obsessive compulsive disorder is not a Mendelian trait in people. It's got more complicated genetics, but it's definitely true that there are different genetic bases, and depending on whether or not you have certain genetic defects that are predisposing for it, you might respond better to certain classes of drugs.
Yeah.
And so that's kind of why it's interesting, because it also then suggested, oh, maybe we should treat the dogs with some of these same drugs that we use for some of the people.
Yeah, because I've heard of dogs getting antidepressants and stuff.
Yeah. We still don't. I don't know enough about the genetics behind the temperament. I don't know how successful antidepressant medication in dogs are, but it doesn't surprise me that many of the medicines work the same. You just have to be careful because again, things that humans can tolerate can be toxic to dogs, and so you don't want to go too far without doing the appropriate testing.
Interesting. Okay. Thank you.
Okay, if there's no further questions, thank you for a completely awesome talk. It was really,
thanks for having me, Adrian. Sorry I couldn't be there in person, but we have the
maybe we should unmute all, and then we'll give you a round of applause.
Thanks.
Okay, that didn't work quite as well as I hoped, but
that's fine.
It was really there.
This has been a production of Cornell University, on the web at cornell.edu.